I.
In
This.
video
We.
will.
Extract.
No, okay, this is boring. Let's try it again. Hi, in this video we extract some DNA from strawberries.
Yes, but what is the DNA?
Yeah, principally the DNA stands for deoxyribonucleic acid and it's the molecule that determines all the characteristics in our body.
That's true. The DNA is formed from two nucleotide filaments. Each of the nucleotides is formed by a sugar and a phosphate molecule, and four azotate bases that are thymine, guanine, adenine, and cytosine.
Yeah, and together with genes are containing chromosomes, and like I said before, they determine all the characteristics in our body.
Okay, let's end the boring part and start the experiment. Now, please, Sofia, tell us what we need for the experiment.
Yeah, okay, so we need a strainer, a pipette, a fork, 2 strawberries, some plastic plates, something graduated, some water, some salt, some dishwashing detergent, and some plastic glasses.
First of all, we need 2 strawberries, and we have to crush them to separate the cell.
Then, for the next step, we need to add some salt to this thing made of strawberries to avoid water from the cell. Then, when we finish to mix, we need to transport it into a plastic glass. It's disgusting.
Okay, now let's add some water and some dishwashing detergent to break down lipids. Wait a second. Okay.
It's really disgusting.
Yes, you are right, Sophie. So now you have to mix it slowly, like this.
Now it's more disgusting. Then we need to filter it to get the liquid part, and when we're done, we need to put it in the graduated glass.
Okay, now we have to add some alcohol to the product that we have and see the results.
I got DNA.
Whoa! Me too! Yes! Look,
It's strange.
these white things are the DNA.
So we got DNA.
Fantastic!
So this was the end of the experiment. We hope you liked the video and
Have a nice day!
Good morning, here we are in an experiment in which we should divide the DNA of a banana from the rest of the banana. So we should start by deciding a fruit. For example, I've decided the banana because it's a good fruit to do this experiment. So we should start by making it become mush. Let's start. Okay, so after you've got a thing similar to this, we should add some salt. we can use, for example, a spoon. And for this quantity, that it is about half of a banana, around half of a spoon is fine. You add it, possibly better than what I've done, and you should, and after you have added, you should continue to make it become mush. Okay, so after we have done this, we should add it in a glass. So after we have put it in a glass, we should add some water. For example, for my quantity, we should arrive around the half of this glass. So I think like this. After we add the water, we should add some dishwashing liquid. So we add it. Okay, so after you have done this, you should put in another glass, and you should filter it using, for example, this thing right here. Okay, so after we have done this, we should add some alcohol. For example, for the quantity that I have, around 1 spoon is fine, for example, and slowly, we should add it slowly. After you have done that, like, just turn it another time, slowly. Okay, so after you have done this, actually from the video you cannot see it, but between there is at the half of of this liquid, there should be the DNA. And nothing. Hope you enjoy it.
Hello, Gabe.
Hello, Professor.
Oh, you don't have to call me a professor anymore. I am the boss of my own company, the DNA & Co.
Congratulations.
Thank you. Now, as you know, we have to finish our last lesson together, and today we will do an experiment about nucleic acids that are
DNA and RNA.
and some. You will need dish soap, water, then salt and alcohol and a colander, and last but not least, a piece of fruit, in my case, a kiwi. First of all, with the help of a spoon, you have to take all the green part of your kiwi, like this, and now, with the help of the spoon and with the help of a knife or of this, you should try to make the kiwi out in smaller pieces. Once you've done with that, you will take a glass and put inside everything you have on your dish. And with the help of the spoon, you can really destroy the kiwi, let's say, so that the cells are more distant from each other, and it will be easier for us. Now that you have this solution here, you will add a little bit of salt, because salt will help us to destroy the proteins that are attached to the DNA. Now you should add a little bit of water, and then you add a little bit of dish soap, because it will destroy all the fats that are in a cell, so that we can access to the nucleus. Now, just mixing up really slowly. Sorry, I think I have a call.
Oh, okay. I'll wait.
Yes? In my company? Yeah. Killer key.
Professor, I don't
What?
hear you very well.
Company.
Is everything okay?
Yeah. Gary, to recall you good.
I don't hear you, Professor. Are you okay? No, I think he's gone. This was the last class, so we will never meet again. I could try to go on with the experiment, not the science, but I can try. We have a colander and alcohol.
The alcohol is needed to separate the DNA from the mixture.
Okay, no,
and
okay,
make
I
it
remember what alcohol is needed for. So now I'll filter this solution so that all the cell parts that are not needed will be taken away. Okay, now that I have this solution, I'll put some in a test tube. Okay, not too much. And then I'll add really slowly some alcohol. Okay, this should be fine. And now I'll wait a few minutes. You should add alcohol in the same amount of the solution. After a few minutes, the DNA has been extracted. As you can see, is this white solid substance in the middle of the alcohol, and we can also take it out of the alcohol. This is our DNA. Okay, now that the experiment is done, I think I should call the professor to tell him. Before, when he was disconnecting, I heard him talking about killer kiwis in his company, but I think I heard wrong. He's not answering me. I'm sure that he was not talking about killer kiwis because they are not real. Oh my god.
Hi everyone, today I'm going to show you how to extract DNA from your saliva. So let's get started. So the materials you are going to use during this experiment are obviously saliva, then a test tube, some dish soap, some alcohol, which is preferable to be cold, some salt, something to count drops with, and something to stir. And that is it. So it's very easy and you can do it with things you have in your kitchen. So before we begin the experiment, it is good to know what DNA actually is. DNA, in fact, is also known as deoxyribonucleic acid, and it's the material that carries all the information about how a living organism works and looks. In fact, it makes us unique in all types of ways. It gives us different eye shapes and eye colors, but it also works on more complex things, such as how your lungs work. The acid is very long and it's contained in chromosomes, which are very tiny as well. Hey everyone, welcome back. It is still me. This is officially the end of the video and the tutorial of the experiment where I extracted DNA from saliva. I hope you found it interesting and learned something new. It is very easy, as you can see. I did it in a few minutes. So I hope you enjoyed it and see you next time. Bye bye.
I today will extract the DNA of a banana. What do you need? A fork, a plate, a banana, salt, a drain, dish soap, water, ananas juice, alcohol, some glasses, a test tube, and a dropper and a syringe. Why we need to smash the banana? You take a piece, only is too much, and then you mash it too. Okay, now that you have this much banana, you'll need to of this soap. So we have it here, more or less like this, and we put it in the banana. You need to stir everything, but carefully. Now you need to 90 milliliters of water, and stay careful and pour it on the side of the glass and not directly on the center. Now that you add water, you need to stir carefully. Now take the drain and use it to and pour the banana in another glass through it. Now you need to take 5 millimeters of what you have done and pour it in the test tube. Then you need to take the ananas juice and take 3 millimeters of it. For then, pour it in, remember on the side of the test tube. Now you will need to take 6 milliliters of alcohol and pour it at the ananas juice on the side from the side of the test tube. If you did the experiment well, there should be a free DNA or this white substance floating in the alcohol. This is because DNA it doesn't dissolve in alcohol.
Good morning, everybody. Today we are going to do an experiment about the separation of the DNA. So, what do we need for this experiment? First, we need some water and some salt that you can normally find in your kitchen, some ethanol, but you can normally use some pink alcohol or the one you use for sanitize your hand, and we need to use some dishwashing liquid, and of course we need a fruit. I decided to use a strawberry, and you can see that I started crushing it, and so let's start with this experiment. So we start by crushing it, as I said before, for separating all the molecules of this fruit. Okay. After that, we are going to put some salt, some kitchen salt. We need to put some salt for like separating all the fat particles and allow the DNA particles to get closer to each other. After we put all the salt, we need to mix it again. So let's do this. Okay. So now what do we have to do? We need to put our mixture in a normal glass. So it's this. You can put it all or a little part. I prefer to put it whole. Okay. So yes. Okay. Now we need to put some dishwashing liquid, but the quantity of the dishwashing liquid depends on the quantity of the fruit, so I'm about to use like half of this liquid. And wait. Okay. And we put some water. We're going to mix it, but slowly, and we start to mix it. We're going to put some more water. And let's continue mixing. So after that, we're going to separate the solid part to the liquid part using this funnel with a filter paper, so that this filter paper will help us to separate it better. And we're going to work with the liquid part. I'm going to put the funnel like this and the filter paper here, and we're going to put all our mixture here. So as you can see, okay, I put it all. So as you can see, the liquid part is going down on here, and the solid part is going to remain here. It will need some time, so I will do this. So I will help the liquid part going down by doing this. Wasn't going down. It's too slow. Okay, it will need a lot of time. Not a lot, but some time. Okay. Now that we wait, we are going to put in this liquid part the ethanol, but not all the ethanol, some like just a little, so that we can see better the separation of the particles. And we are going to see, personally, that the DNA is going to be at the top and the liquid part. so we wait. I decided to remove the filter paper because it was kind of slow. So like you can see now that it's going down faster. So we're gonna take it off. Being careful. Okay, I'm gonna put it here. Okay. Hoping I'm not gonna break. Okay. So I'm about to put some ethanol in here. We're doing like this because if it goes down too fast, we can damage all the experiments. So we're going to let it go on the walls. Okay. And now we actually have to wait until the experiment is done. Hoping it worked. As you can see, zoom it. You can see here that there's a little separation. It's not at all clear, but if you come really near to the beaker, to the glass, you can see it. So that is the experiment. There's like a less colored part that is the DNA in here, and other part of the liquid is under here. And we're done. This is the experiment. For seeing better this separation, I decided to put this liquid into this little glass. So I'm gonna go up here so I won't make. So I hope it works. So let's wait again to see the separation. As you can see here above, there are the histones, because if we have put the ananas juice, these histones wouldn't be there, but I didn't put it, so here are histones. Okay, so I decided to put some more ethanol, and now you can see that there are more histones, so the separation is happening again. So actually need to wait. In fact, if you go here, you can see that there's a little separation of the DNA, but there are too many histones, so it's kinda hide. But if you get closer, here is the separation of the DNA.
Everyone, today I'm here to extract the DNA from a kiwi. The kiwi is made for you choosing one. So what we need is very simple. You can find in your house, and let's see what we need. We need dish soap for your dishwasher, alcohol, preferable cold, then pineapple juice, a test tube, water, normal water, a strainer, 2 glasses. This is a wooden stick, and this. Okay, so let's start. In the first step, we take the kiwi, we cut it outside, and we smash it a lot because we have to separate the particles of this kiwi. This is what it should look like. In this step, we have to. So we transfer the thing we did inside of a glass. We can smash it only because we need it the most smashed you can. This is what it should look like. Then we add water and salt, and we mix it a bit. In this step, we put what we did with the strainer in a new glass. It's very important, this strainer. It should look like water. In this step, we have to put the dish washer, the dish soap inside of it. 4 or 5 bunches, what you want. I put 4. It's very important. In this step, we have to put the pineapple juice inside of it. So, not so much, a bit, like that. And we mix it very gentle, not to make bubbles. It's very important not to make bubbles. This is the last step, and we have to put what we did inside of the test tube, and we have to put the alcohol, very cold alcohol, inside of it with the pipette. So let's try to separate the DNA from the other things. So this is what went out to me, is DNA, I think, and is this thing here. So this is the DNA. You have to try it absolutely at home. Goodbye!